In one hospital room, an oncologist is intentionally poisoning the well by injecting a specific therapeutic enzyme. They wipe every trace of asparagine from the bloodstream. The malignant cells, entirely dependent on scavenging this nutrient, rapidly starve and collapse. Down the hall in the neonatal intensive care unit, a newborn is suffering from intractable seizures. This infant inherited a genetic defect that prevents their developing brain from manufacturing that exact same molecule. While the oncologist destroys it to save a life, this infant's brain is structurally failing because it lacks it. Between these two extremes sits a highly sensitive diagnostic lab. If the blood sample drawn from either patient sits on a counter at room temperature for just 30 minutes, the molecular evidence will literally degrade in the tube, feeding a false reading back to the clinical teams. This incredibly fragile molecule is L-asparagine. Its unique nitrogen-dense amide group functions as a highly specific structural alignment node, stabilizing the three-dimensional folds of circulating proteins. This configuration creates a hydration shell that prevents these proteins from aggregating into useless, tangled masses inside your bloodstream. The precise handling of this single polar molecule determines whether the human physiological architecture successfully constructs itself or undergoes a catastrophic structural collapse. Scientists first isolated asparagine back in 1806 by purifying asparagus juice, making it the very first amino acid ever discovered. At the physiological pH of 7.4, asparagine operates as a net neutral polar zwitterion. Because its side chain lacks an ionizable charge, it is physically incapable of driving general acid-base catalysis or trapping metals. Instead, the body uses it for pure structural stability. Roughly 60% of your total asparagine supply is locked away in the extracellular matrix, heavily woven into the cell surface receptors and circulating immunoglobulins that connect your tissues. The tiny fraction of free asparagine circulating in your blood plasma is merely a fragile temporary transport phase. It is constantly being pulled from the vascular space to supply this massive life-sustaining network. To see exactly how cells use this supply, we have to look inside the rough endoplasmic reticulum, where a mechanism known as the sugar clock operates. Here, asparagine serves as the absolute primary anchor for a process called N-linked glycosylation. When the cell's machinery reads a highly specific genetic sequence, asparagine followed by any amino acid, then serine or threonine, an enzyme complex bonds a preformed corm carbohydrate chain directly onto the nitrogen atom of the asparagine anchor. Once attached, enzymes progressively trim glucose residues away from this sugar chain, piece by piece. Chaperone proteins continuously read the remaining sugars on this shrinking chain, using it as a literal timer to verify that the newly synthesized protein has achieved its correct three-dimensional shape before the clock runs out. Beyond timing protein construction, the molecule takes on a second role at the cell border, functioning as a microscopic currency. When a cell wants to grow and divide rapidly, it needs large neutral amino acids like leucine from the outside environment. To get them, the cell pushes its own internally manufactured asparagine outward through a specialized revolving door called the ASCT2 antiporter. This outward push pays the toll, pulling leucine inside, which then directly activates the mTORC1 pathway, the anabolic switch for cellular growth. By tying these two mechanisms together, the cell ensures it only initiates global replication when it has enough internal asparagine to successfully fold the massive amount of new proteins that division requires. This system breaks down completely when cells lack the enzyme asparagine synthetase, making them unable to manufacture their own supply. In newborns with the genetic defect asparagine synthetase deficiency, the developing brain completely lacks this vital enzyme. You might assume doctors could just run an IV line to replace the missing nutrient, but the blood-brain barrier simply lacks high-capacity transport loops for asparagine. You can flood the baby's bloodstream with therapeutic supplements, but the nutrients will bounce right off the barrier, leaving the neurons inside to starve and trigger severe microcephaly. Acute lymphoblastic leukemia cells share this exact same metabolic blind spot. These rapidly dividing malignant cells also have zero internal production capacity. To survive, they are forced to scavenge free circulating asparagine directly from the patient's blood plasma. Oncologists exploit this vulnerability. By injecting the therapeutic enzyme L-asparagine, they rapidly hydrolyze and destroy the free vascular asparagine pool, selectively starving the malignant cells of the very material they need to build new proteins. The collateral damage of this strike is immense. Destroying that much asparagine at once dumps a massive load of free ammonia directly into the patient's vascular system. If the liver's processing networks become saturated, this highly toxic ammonia crosses into the brain, causing hyperammonemic encephalopathy and involuntary loss of motor control. Manipulating this specific amino acid in a clinical setting requires flying inches off the ground. The medical team has to carefully manage the toxicity, balancing between starving a lethal disease and inadvertently poisoning the human host. You don't have to be in an intensive care unit to witness the physical alteration of this molecule. The same structural breakdown happens under extreme environmental thermodynamics. Starchy plants like potatoes and cereal grains naturally contain high concentrations of free asparagine. If you boil them, they remain stable, but when you expose these starches to high, dry heat, anything above 120 degrees Celsius, like when you drop them into a deep fryer, a violent chemical synthesis occurs. Driven by the intense thermal energy of the Maillard reaction, the free asparagine condenses with ambient glucose sugars. The heat forces the molecular structure to break and reform into a completely different compound altogether, acrylamide. The exact same molecule required to successfully build a developing fetal brain physically transforms into a volatile neurotoxin and suspected human carcinogen, all because of 3 minutes in hot oil. Back in the hospital, monitoring the precise vascular levels of this molecule is a matter of life and death for the leukemia patient trying to avoid ammonia toxicity. To get that exact number, clinical labs rely on liquid chromatography tandem mass spectrometry. This multi-million-dollar technology is required to accurately separate asparagine's specific mass-to-charge ratio from other closely related complexes, like basic aspartate. Yet all of this heavy analytical artillery is completely vulnerable to a simple human error known as ex vivo deamination. Human blood naturally contains active enzymes. If a drawn blood tube sits on a counter at room temperature for just 30 minutes, those enzymes will literally chop the amide group off the free asparagine, artificially converting it into aspartate right there in the tube. The most advanced mass spectrometer in the world will accurately read what is present in the sample. But if the phlebotomist fails to slam that tube onto crushed ice immediately after the draw, the machine will report a false critical low, potentially sending the clinical oncology team into an absolute panic over a metabolic crisis that never actually happened. Because of these pre-analytical dangers and a tight total allowable error of just 15%, measuring asparagine is an operation requiring strict diagnostic stewardship. This test is explicitly reserved for high-stakes leukemia management and rare neonatal screening, not for working up a patient with general fatigue. Asparagine integrates structural construction with metabolic signaling. It acts as the timer in the endoplasmic reticulum's sugar clock, operates as the cellular currency to trigger replication, and serves as a vital substrate for neurodevelopment. The tragedy of asparagine synthetase deficiency reveals a limitation of systemic biology. Having an abundance of a nutrient in your blood means nothing if the physiological architecture of the blood-brain barrier prevents it from reaching the starving tissue. The body maintains its physical architecture by constantly regulating these microscopic anchors. One molecule at a time, this system prevents the structural collapse of the extracellular matrix, guarding the stability of proteins in your blood.